Hello, ladies and gentlemen. This is Dan Montano. I want to welcome all of you to our presentation on Parkinson's disease. Is therapeutic angiogenesis a treatment or even a possible cure? I know that everybody is really interested in hearing from Dr. Jacobs and not me, so I won't belabor the point. I am Daniel Montano. For those of you that are new, I'm the Chief Executive Officer of Zhittya Genesis Medicine. First of all, for a lot of you out there, there's been a great deal of confusion with our monthly update. For those of you that are old timers with us, you know we try to get an update out every month. Our list has gotten so big, we have over 100,000 readers now, I believe. And so we went to a new mailing system in January. And as you can see, we've had tremendous problems getting the notices out to everybody and getting the information out. So if you're one of our subscribers and you did not get an update, please contact myself, Vika, or zgm.care and resubscribe. We have to repair things. And if you're new, we'll talk about how you can sign up for our updates. And we use our updates to discuss our medicine, where we are, where the clinical trials are. And I believe it's relatively informative. Now, before we turn over to Jack, I just want to explain that this has been a chaotic couple of months because we've been extremely busy. Ever since we uncovered the fact that the intranasal may be a breakthrough way to deliver our medicine and maybe a breakthrough way to treat Parkinson's disease, we've been super active advancing that. Along the way, we developed these compassionate clinical trials, and everything has been going amazingly quick. Now, I believe that we have about 100 doctors on this webinar, and that is probably the result of webinars that Dr. Jacobs has done in joint partnership with people like Dr. Shahidi of Morocco, Dr. Al-Anti of Riyadh, Saudi Arabia, Dr. Salek of Jeddah, Saudi Arabia, and Dr. Omar Halek. And these people have done Zoom webinars with Jack on brain, heart, other issues, and we've developed an amazingly response. And we have, as I said, I think over 100 doctors on this call, and I think it's going to be great that they're looking at this new treatment. Now, when I say we have a lot of people on, based on my last glance at who signed up, we have doctors from Morocco, Algeria, South Africa, South Korea, Azerbaijani, Algeria, Iraq, Mexico, Senegal, Indonesia, and many more. So for the people in the audience, we're going to be talking about our medicine, but also along the way, we're going to be talking about our desire to do business with clinics out there and expand this so that we can take on more people. And I'll discuss that a little bit later on. But the important thing is that if you're in a country that's not heavily regulated, like the United States is, the clearance problem is done by medical doctors, not bureaucrats. We want to talk with you. We know that there's a tremendous amount of opportunities out there to do what we believe are compassionate trials and get clearance in countries all over the world. So if you're a doctor in Baku, Azerbaijani on this call, and you're in the center of an area that 100 million people around you, we'd love to explore setting up a clinic with you, doing a clinical trial with you, and seeing if our medicine works in Azerbaijan as well as it works in the United States or the Bahamas, and treating with your clinic, letting you be the center that treats the people in that area. I believe the idea that we have 100,000 subscribers and you have 100 million people in your area, and based on my crude calculations, I'm going to say 300,000 people that suffer with Parkinson's, which means this could be a huge addition to your medical practice. Now, before I go any further, I'm going to turn this over to Dr. Jacobs. I want to make the following comment that we are working on the clinical trials, compassionate use, and Dr. Jacobs is going to explain to you first our medicine, why it works. Then we're going to have a question and answer period. And after the question and answer period, we're going to have a short summary by me. And then we hope to hear from you people again. I'd like to introduce Dr. Jacobs. He's the one on the screen. That's him waving his hand. He's a brilliant scientist, a wonderful human being, and my partner for 24 years, which is either a sign of his tremendous patience and tolerance or his lack of being able to find other friends. Dr. Jacobs, please give the presentation.
Okay. Thank you for that introduction, Dan. There are a lot of new people on this webinar who haven't heard our story before. So just to catch everyone up and for us all to be on the same page, I will go through some things that some of you have already heard, but I want to talk about how our drug works, how it stimulates angiogenesis, which is simply growing new blood vessels in damaged tissues. Talk about our drug, talk about just one clinical trial that was done here in the US, injecting this drug into heart patients, because we've got a lot of safety data from that. And then I'll go in and talk about Parkinson's disease, results in animal trials, and then talk about our planned clinical trials in Mexico and the Caribbean. Okay, let's talk about angiogenesis. Angio is blood vessel, genesis is new, so angiogenesis is a process in our body where we grow new blood vessels to heal cuts and scrapes. And I'll talk about therapeutic angiogenesis, which we stimulate with our drug candidate. So this skateboarder scraped his arm, fell off a skateboard, and he's going to use this process of angiogenesis to heal this wound. We zoom in on his scrape there. So this is a wounded area of skin. This is a scab over that wounded area. We look underneath that scab and you can see all these brand new blood vessels stimulated by our drug, which I'll talk about in a second. But these blood vessels, new blood vessels, bring in blood perfusion to heal, to regenerate skin here in this case, sweat glands, nerves, hair follicles, whatever you need to have skin as an organ. And again, this process of angiogenesis is stimulated mainly by this protein, which is our drug candidate, human fibroblast growth factor 1. It's a protein, as shown here. It's like insulin or human growth hormone. It's not messenger RNA. It's not gene therapy. It's not stem cell. It's a natural growth factor that is a potent stimulator of angiogenesis. And in damaged areas of the brain, this process of angiogenesis is linked very tightly to neurogenesis. So we have those two processes going on in the brain, and I'll talk about that in a second. I was actually involved in purifying this protein from cow brains as a graduate student. So I've been very interested in this protein for many years. We don't use cow brain FGF1 in the clinical trials. We make the human FGF1. We use what's called recombinant DNA technology. We make it in an FDA inspected facility as shown here. So the FGF1 we're using for the human clinical trials is pure, it's potent, it's sterile. And this went a long way to making clinics in the Caribbean, clinics in Mexico feel comfortable about using our drug in clinical trials. Okay, let me just briefly talk about the natural process of angiogenesis. When that skateboarder scraped his elbow, his body immediately made FGF1, as shown here. And that goes and binds to the FGF1 receptor, shown in yellow here, these lined blood vessels. And that starts, that's kind of a key going into a lock here, that initiates this whole process of angiogenesis. So we see these endothelial cells migrating and proliferating. And here, the very first sense of a new blood vessel, this is a tube. It elongates and brings blood perfusion into that wounded area. Now, one important point that's shown on the slide is that when there is damage or injury to a tissue, we get an upregulation, increase in number of these FGF1 receptors. And this is very important. This gives us a nice safety window. So I often get questions, if we inject FGF1, do you get blood vessels growing in other parts of the body? And no, we do not. We do not see that. Only in damaged or ischemic tissue do you see new blood vessels formed. And so that's a very important point. Just one clinical trial I'll talk about. This is one that I supervised. This is in patients who had severe coronary artery disease. They couldn't have a bypass. They couldn't have a stent put in. And ABC News, I'm going to show you just a 1-minute news clip. They went down to one of our 6 clinical trial sites. It was at the University of Cincinnati. And they interviewed the cardiologist there, Dr. Lynn Wagner, who was conducting the study, as well as a few of the patients. So let's watch this, and I'll come back after the clip is over.
But since receiving an experimental treatment for his blocked arteries, his pain is gone.
I really feel great.
Duke was one of the first heart patients in the country to be treated with a protein actually capable of growing brand new arteries. The genetically engineered protein is injected directly into the heart. Within days, a network of new vessels begins to grow around the blockage, increasing the blood supply. Dr. Lynn Wagner showed us the changes in one patient's heart.
We see a small, narrow main artery and not very many secondary and tertiary arteries. This is after the treatment. What we're now seeing is new blood vessels growing here off the end of this artery.
And the patients themselves?
Symptomatically, they're improved within a couple of weeks of the treatment.
Just ask Constance Donnelly.
Oh, I feel wonderful. I've never felt so good in the last 5 years.
But doctors already see potential in other cases where the blood supply needs a boost, such as strokes and diabetes.
So that woman there, the patient Constance Donnelly, came into that trial in a wheelchair. She was a cardiac cripple, and you can see that was 12 weeks after she was injected with the FGF1, and she was up and about. So we'll be continuing those heart trials, and there was about 90 patients who got FGF1 into their hearts, and we looked very carefully, blood vessel growth in other parts of the body, in the eye. We took photographs of the retina. We saw no adverse events from the stroke treatment. Okay, let's move into the brain. As many of you know, the brain is incredibly vascularized, probably the most highly vascularized organ in our body. Billions of capillaries feeding billions of neurons. So let's just take a little bit closer look at that. This concept of the neurovascular unit. So we have these large arteries in the brain branching down to smaller arterioles and then branching down to the smallest blood vessel in the brain, the capillary. So this is a capillary shown here. And this capillary, basically every neuron has its own dedicated blood supply, bring nutrients to the neuron, remove metabolic waste products. And also these capillaries, it's not shown on this slide, but there are neural stem cells distributed throughout the brain that need an adequate blood supply to be active. And we'll talk more about that at the end of the talk. But if this blood vessel gets damaged by atherosclerosis, other toxins, neurons adjacent to those blood vessels are going to suffer. And as I mentioned previously, our neural stem cells need blood to divide and mature. And again, we'll talk about that more at the end of this talk. Okay, let's now talk about Parkinson's disease, evidence that we and others believe this results from blood perfusion defects, dysfunctional blood vessels in the brain. I'll then talk a bit about some of the animal studies that have been done with FGF1 in animal models of Parkinson's. And then I'll go on to talk about the upcoming clinical trials, both in Mexico and in the Caribbean. As most of you are aware, Parkinson's disease results from a decline of functional dopamine neurons. We're born with about 500,000 dopamine neurons. And as we age, just in healthy aging, we lose a number of those dopamine neurons, but not to the extent that someone with Parkinson's disease does. So here in blue is this inexorable loss of dopamine neurons in a Parkinson's disease patient. When they've lost about 60 to 80% of their neurons, they begin developing the symptoms of Parkinson's disease. So in our clinical trials, we'll be treating patients and trying to increase the number of dopamine neurons, as I'll show you we did in monkeys, to areas where they're become asymptomatic. So the dopamine neurons are located deep in our brains. They're actually pigmented neurons. They're dark neurons. Here they are here. And when they become dysfunctional, we believe due to a lack of blood flow in this region, less dopamine is secreted and movement disorders ensue. Now, this is not our work, but I'm going to show you on the next slide. You can use functional imaging, functional MRI, to actually quantitate blood flow in this area of the brain. And we can look at both normal individuals and individuals with Parkinson's. So this is using this imaging functional MRI. Here's a healthy 21-year-old. So this is blood perfusion in that area, the substantia nigra region of the brain, which houses the dopamine neurons. And this healthy 21-year-old has full maximal 100% blood flow. As we saw earlier, as we get older, this is a healthy 65-year-old, about a 25% decrease in blood flow in that small region of the brain. But here's an individual with Parkinson's disease, 50% reduction in blood perfusion in that area of the brain which houses the dopamine neurons. So we and others believe this lack of blood flow, lack of blood perfusion, is basically choking off these neurons and making them dysfunctional. We know that patients with Parkinson's not only have movement disorders, but also can develop other symptoms, loss of smell, loss of speech, dementia. And here's another one of those functional MRI scans showing blood flow. This is in a woman who had Parkinson's disease plus dementia. And here we're looking basically at the whole top of the brain. This is the cortex region of the brain. And this is where memory functions occur, emotional, executive functioning. And in blue, a healthy 21-year-old, this whole area would be blue, normal blood perfusion. But in this patient, we see in yellow, that's decreased blood flow, and red is severely decreased blood flow in these regions of the brain. Again, they're lining up with regions that control memory and executive functioning. So we would predict and hope that if we gave a patient with Parkinson's FGF1, and the FGF1 got throughout the brain, which I'll show you it does, that we not only could improve their Parkinson's disease, but hopefully also improve their cognitive abilities. Okay, let me briefly show you some data on two models of Parkinson's disease. One, and these are the two main models researchers use before drugs candidates go into the clinic. There's a rat model of Parkinson's disease where a neurotoxin is injected into the brains of these animals, and they come down with Parkinson's disease. And this just shows that our drug, FGF1, this was given intravenously to the rat, can stimulate regeneration of new dopamine neurons in the brains of these rodents. So new neurons stain brown. These are new dopamine neurons in the FGF1 treated animals. They also have an improvement in their motor scores. Where in the placebo-treated animals, we don't see that regeneration of dopamine neurons. But the gold standard for testing drugs before the clinic is in this primate, in this monkey model of Parkinson's disease. And just as is done with the rodents, if you inject a neurotoxin into the brains of these animals, after a number of months, they come down with all the classical motor symptoms of Parkinson's, tremor, gait disturbances. And so FGF1 was tested in this animal model and performed very well, actually. I'm going to show you first the improvement in motor scores in monkeys with Parkinson's disease. This is a motor score. 4 is normal movement, normal motor score. 1 is highly abnormal. And you can see after the toxin is injected, 9 months are waited. You can see these two groups of monkeys. One's going to get a placebo dose, and this group in blue will get an FGF1 infusion. And here we see, as we infuse FGF1, a gradual improvement in these motor scores. The experiment was stopped here, but perhaps if it continued, these monkeys might have returned to normal. We don't see that in the placebo-treated animals. They continue with their highly abnormal movement. Now, importantly, we can look in the brains of these animals and see, as in the rats, are we regenerating dopamine neurons with FGF1 treatment? And indeed we are. Again, similar to the rats, you can stain newly regenerated dopamine neurons in brown here. These are the FGF1 treated animals. You don't see any of that regeneration in the placebo treated monkeys. So in monkeys anyway, we feel we're tackling one of the main root causes of Parkinson's disease, that is loss of these dopamine neurons. And the second pathology we can look at in the monkeys is these aggregated tangles. This is similar to beta amyloid plaque that is seen in Alzheimer's disease. In Parkinson's disease, patients get these toxic tangles of alpha-synuclein protein, as shown here. And we can see after treatment with FGF1, we can basically wash away this toxic protein, not quite back to normal, but certainly better than this situation. We believe by addressing these two root causes of Parkinson's, regenerating dopamine neurons, getting rid of this toxic tangle protein, that leads to the motor skill improvement in these monkeys. And so this data was submitted to a number of regulatory bodies. And let me talk now. We first submitted it to our FDA, which we always do, and they thought the clinical trial was fine, but they wanted a bit more animal toxicity data, which I felt we had plenty of, but we don't question our FDA, so that those animal trials will be done. The FDA has approved the studies that we proposed, but we submitted the same package to Mexico, and they said, this looks wonderful, please start. So that trial in Mexico was, the design of that was administering the drug intravenously. And we kind of got held up a bit in Mexico with COVID-19, but we plan to be starting there soon. It's been approved to treat patients. We're going to study 3 rising doses of the drug. No placebo group. All patients will receive a 1-hour intravenous infusion of FGF1 5 days a week for 4 weeks. So it's 4 weeks of treatment. And we're going to administer the FGF1 through a PICC line, which is a central line. It's a port, basically a port that stays in place for the entire 4-week clinical trial. And that's how the drug will be administered, and blood taken out for lab tests, things like that. Now, the regulatory body in Mexico wanted us to follow these patients for a year. So they'll be, after the 4 weeks of treatment, they'll be coming back every couple of weeks to have safety testing done. Now, as Dan mentioned, we recently got some very nice results back. We have a collaborative research going on with a research group in Minneapolis, Minnesota, who were pioneers in delivering protein drugs into the brain through the intranasal route. And they've actually have done a number of clinical trials. Their field is delivering insulin into the brains, and they're looking to treat Alzheimer's disease. We sent them some FGF1, they made it radioactive, and they did an experiment looking at how much of our FGF1 got into the brain through the nasal delivery route or through the intravenous route. So we could compare the two directly. After they administered the FGF1, they dissected out various regions of the brain, as shown here. And since they knew we were interested in Parkinson's disease, they actually dissected out the substantia nigra region of the animal's brain. So here are the results. That's a busy slide, but let me walk you through it. Basically, what they found is that FGF1 was widely distributed throughout all areas of the brain after intranasal delivery. They compared, let's look here, they compared this is intranasal delivery in one formulation, this is intranasal delivery in a second formulation, and this is intravenous delivery, as shown here. So basically, throughout the brain, here's the substantia nigra, about twice the amount of FGF1 got into the brain, into this area of the brain, when you compared intranasal versus IV. And that pretty much held up throughout the brain. Here we see intranasal versus IV, intranasal versus IV. Let's focus in a bit, since we're talking about Parkinson's disease. Let's focus in on the substantia nigra region of the brain. So this was taken from that previous slide, but here's intranasal delivery. This is the concentration of FGF1 in that area of the brain. It's nanomolar for any scientists who are out there. And the important point I want to make on this slide is we know how much FGF1 has to be in the substantia nigra to fully activate all the FGF1 receptors to get angiogenesis and hopefully neurogenesis. And that quantity is 0.04 nanomolar. Be about at this region of the graph, all right? So once we get above that region, all of these FGF1 receptors would be activated, and we think we would have a very robust angiogenesis and neurogenesis response, whether we give the drug intranasally or whether we give the drug intravenously. So we feel both in Mexico and in these trials I'm going to be talking about now in the Caribbean, we should see a therapeutic effect and hopefully benefit from FGF1 treatment. There's an intranasal delivery device, which is approved for use in humans. This is the one being used in the insulin studies with Alzheimer's disease patients. It's made in Seattle, Washington. We have a contract with the manufacturer to supply us in our clinical trials. So the FGF1 is sitting inside here in a reservoir. There's a small motor in here, and it dispenses the FGF1 through here after the patient or a nurse depresses this button. So it's a fairly simple device. What it does, though, why it's essential, is that it atomizes the FGF1. So the droplets that come out are very, very tiny, and it allows it to get to the upper part of the nasal cavity where you need to put it for it to get into the brain. And one other point I want to make is that in the trial, we'll just be delivering this 2 snorts, maybe after breakfast, once a day. So it's a very simple delivery. All right, let me talk about this first initial clinical trial we're be doing in patients with Parkinson's disease using this nasal inhaler. Regulatory bodies like you to start at a lower dose just to see if there's any safety issues. Remember, we've never studied nasal delivery in humans with FGF1, so we'll start at a low dose for 2 weeks. Every day, the patient will administer, well, a nurse will administer this first trial, FGF1 intranasally up the nose once a day. 3rd week, the patients will come to the clinic for safety testing, vital signs, lab tests, which we routinely do in our other clinical trials, monitoring of the heart. And then there's a very simple Parkinson's disease questionnaire, which can be administered, which is validated to use in clinical trials to test the efficacy of a particular drug candidate. We'll then go to week 4, where we'll give them what we approximate the dose the monkeys got to reverse their Parkinson's. So it'll be a higher dose, double the low dose. And then only for this first clinical trial, will we keep the patients for 3 months. That's the follow-up with weekly visits. And very importantly, we're going to be rehabilitating these patients during this period of time. Even if we grow new neurons in the brains of patients, they probably will still need rehabilitation to, say, learn to speak again or learn to walk properly again. So that we feel is a very important concept. We're not going to do this in our subsequent clinical trials. People can come down. We might, in the second trial, watch them for 30 days, and then later on, perhaps there will be no observation period needed when we know this drug is safe and that it works. Clinical trials in the Caribbean, we've identified clinics in 3 island nations to date: the Bahamas, Cayman Islands, and British Virgin Islands. We're first submitting a clinical trial to conduct in Parkinson's disease patient intranasally, and then we hope to follow that up with an intranasal application in ALS or motor neuron disease. And I'll actually be talking about this clinical trial in a week's time in another Zoom webinar. If you haven't seen enough of me, you can dial into that. And then actually the clinic in the Bahamas has 2 operating suites for heart patients. So they are interested in exploring doing a no option heart disease trial, much like the one I showed you the video on. The initial first trial will be 3 months long, as I mentioned, to test safety and efficacy. And our current plans are to kick that off in April of this year. Once we see that it's safe and we see some efficacy, we would greatly expand subsequent clinical trials. And we obviously would work with the clinics that we've contracted with, what their flow through rate could be, and see what they're comfortable with. But we hope to greatly expand the studies at those sites. We're also in discussion with other countries, including the ones you see down here at the bottom of the screen. We hope to carry out trials in those countries as well. Okay, doing these clinical trials, I think is going to give us a really good opportunity to look at subgroups of patients with different symptoms or different treatment modalities with Parkinson's disease. And so, for example, after the initial clinical trials, if we see our drug is safe in the people who are in that initial clinical trial, we can do additional small clinical trials in Parkinson's patients that, say, have deep brain stimulators in and working, or in patients who have had DBS, but the DBS has been removed because it's become ineffective. 40% of patients with Parkinson's have depression. We could combine, we could do a subgroup with speech issues, dementia, balance issues. So I think we're going to learn a lot about how well our drug works in Parkinson's disease, but also, Parkinson's is a very diverse symptomatic disease, so we can look at these individual symptoms in separate clinical trials. Of course, after we start up the Parkinson's and ALS, and we see FGF1 is safe in the brain, there's no reason we can't pursue other clinical trials in these clinics. Stroke disability is one that we've targeted. So that's after a person has a stroke, is stabilized, but has a disability. Multiple sclerosis, a lot of people who have contacted us with interest in that. obviously Alzheimer's disease and dementia. And one thing we haven't talked much about, but we have an interest in maybe after we begin treatment of these various diseases, is the aging brain. And there's a lot of interest that dysfunctional problems as we age, forgetting names, forgetting car keys, is a vascular problem, is a vascular issue. And I want to show you just quickly to end my talk, 2 studies which indicate that as we age, our brains perform worse because our vascular system is breaking down, either getting clogged or leaking. And I think if we have a drug that can reverse that, we have a potential therapy for age-related cognitive decline. And especially if we have a intranasal delivery system, that's something that can be easily administered to patients as they age. Okay, let me show you this first study. Is from Kremble Neuroscience Center in Toronto, Canada. And they used imaging technology to look in real time. It's pretty remarkable. You see, they actually could see mini strokes occurring in real time in the aging brain. And the patient didn't know they were having a stroke. The caregiver, the doctor didn't know they were having a stroke, but the strokes were occurring. Okay, so they had 5 adults who they recruited who had white matter disease in their brains. And they actually scanned them once a week, every week for 16 weeks, again, with an imaging modality that could look at tiny strokes. So here, this is this white matter disease they have. And then they started looking, you're going to see new imaging technology, which can look at strokes over a 15-week period. There's one, see there's a little tiny stroke, another one up there. This is mini strokes happening in real time. There's another one. You'll see one up here. Yep, there's another mini stroke. So what these authors in Canada said is that we all have these mini strokes, me, you, and they occur and they accumulate over the years. And in this particular group of patients, they developed white matter disease. But they also speculated that if they happened in cognitive areas of the brain, memory areas, they could affect cognition. So again, this is vascular dysfunction, vascular disease. This is what our drug FGF1 can treat. The second study is from Columbia University Medical School on why our brains perform worse as we age. Some of you have seen this already, but for those who have not, let me give you the punchline. So the researchers there at Columbia in New York City, their hypothesis was that a lack of blood flow in the brain as we age makes these neural stem cells less active. So young people and old people have the same number of these neural stem cells, but what is different in the older people is that they don't have the blood perfusion to keep these cells active and dividing. So for example, in an older person with Parkinson's disease, there would not be enough blood flow to get these neural stem cells to differentiate into the dopamine producing neurons that are needed. And again, this is about a 1-minute video, so let me run it for you, and I'll be back afterwards. So we agree completely with that video. I've talked more about diseases of the brain, but if we can improve blood flow to the aging brain, I think we could help with these cognitive issues. So I want to thank you for your attention. We do have a very simple website, zgm.care. If you want to participate in these clinical trials, you want more information on our company, if you want to see me more, I have about 13 45-minute videos on this website, so you can go there. And you can contact us too. Here's emails for Vika Montana, will come on in a second for the question and answer period, and then Dan, who you heard from in the beginning. So again, thank you for your attention, and we can open this up to questions and answers.
So we have a lot of questions. I've been answering some of them in writing, but here's some that will be answered on live on air. First question is to Dr. Jacobs. I think it's the best question to you. You said that there will be two ways to introduce the drug into the brain. One will be by IV route and second one by intranasal route. Why the two ways?
Okay, that's a good question. It really was a matter of timing. When we first submitted our clinical trial to Mexico, we had no idea the intranasal route would work or even existed for that matter. So we went in, and the only way we thought we could get it into the brain, and that it was intravenously. Since that time, we've learned that we can get more FGF1 into the brain by intranasal delivery, and that's why we're now pursuing those clinical trials in the Caribbean and in those other countries I mentioned. We think both methods will work. As I talked about, we think we get enough into the brain by both methods to work either intravenously or intranasally. And quite frankly, we're very curious to see which method brings better relief to the Parkinson's disease patient. So that's a very important consideration as well. So that's why we're carrying forth with both forms of administration.
There's another one. I suffer from multiple sclerosis. When do you plan to start clinical trials for this disease?
Yeah, that is a good question too. Again, I kind of gave the order. We were going to start the Parkinson's first, followed hopefully quickly by ALS, a motor neuron disease, or Lou Gehrig's disease. And then the next trial we would pursue in those compassionate use clinics, whichever ones we have would be studies in multiple sclerosis. So we know we have a lot of our people who follow us are interested in MS, and we plan to do that trial probably as the third clinical trial in those regions.
This question is for Daniel Montano. This question comes from Ali. I'm a Parkinson's disease sufferer. I'm also an investor in Zhittya Marketing Partner, Regenerative Medicine of Middle East North Africa, MENA. In January update, I read about a new company being created to advance these intranasal clinical trials, Zhittya Intranasal Development Corporation, or as you refer to it in update as ZINC. How can I invest in that project? I want to advance this treatment for Parkinson's disease, and I want to make money also. If this works, I believe it will be big.
Well, first of all, thank you, Ali. I'd like to say that, as you know, we like to take care of our investors. There's nothing better than happy investors, and we believe that we do our best to do that. In answer to your question, this is really a webinar for the medicine and for the public, and I don't like to talk business or money in public. So if you're interested in either learning more about the investment opportunity or setting up a clinic or a business, email me personally at dan333@aol.com. That's my personal email address. We'll be sending that out with a notice. And in that case, we'll have a personal conversation. The good thing about business and money is that's my favorite topic, and I like to talk about them in private. But that's the way it should be done. So just send me an email, okay?
So this question is to Dr. Jacobs. If the dosing is only for 4 weeks, why are you requiring people to stay 12 weeks?
So you can enjoy the wonderful beaches in the Bahamas. No, I think I sort of answered that question in my talk. So for the initial clinical trial, again, this is the first time FGF1 has been delivered to the brain in humans through the intranasal route. We want to check carefully for any adverse events that might crop up. And so we'll be checking that for that 12-week period in that initial group that comes to the Caribbean. After that, people will not have to stay in the Caribbean unless they want to for 12 weeks. But we hope to decrease that to 30 days. And then probably, if we've been dosing patients for a year and it's safe and it looks like it's working, there's no reason they have to stay in the Caribbean. They could get their medicine and return to their home countries and administer the drug there.
This question is to Dan Montano. This question is from Jim. I heard the public share stock offering for the shares to trade on the New York Stock Exchange is happening soon. An IPO investment would be of interest to me. How I can learn more?
Thank you very much for the question. To be very candid with you, the United States Securities and Exchange Commission has extremely strict rules about when and what you can say about going public. I was an investment banker from 1968 till 1998. So for 30 years, I was in this investment banking business. So I know these rules very well. We're not going to say anything until the lawyers give us what we can say and the lawyers tell us when we can say it. And when that happens, we'll post it on our website. We'll post it on the YouTube channel, and we'll send it out to all of our update people so you can take a look and decide for yourself. But I would once again make the comment that I talk about money in private. I don't talk about it in public. But thank you anyway, Jim.
So this is a question that many people ask, what side effects of adverse events have you seen with FGF1?
Most of the human safety data has come from the heart trials that were done with FGF1. Again, we looked carefully, we took retinal photography. So are there new blood vessels growing where they shouldn't be in the retina? We saw no adverse events in the heart trials that were directly attributed to the FGF1. We looked at tumor markers. People often ask us about cancer. We followed that closely. But let me just say that this drug, this growth factor, has been out there for a long time. Merck originally developed it, and they've published a lot of data, both in animals and toxicity data. So in the published data, there were no adverse events seen in their animal trials. And so to date, we really think this is a safe growth factor. We have obviously certain exclusion criteria. We'd never give this to a person who had active cancer or had other issues like in their eyes, any retinopathies, but we'll screen patients carefully before they treat them. But this is a pretty safe growth factor in my humble estimation.
I just wanted to remind you a little fact, Dr. Jacobs, that pregnant women have high dosage of this drug in their system. Is it correct?
That's right. Thank you, Vika. Yeah, Vika Montano actually found this, papers on this research. So when a woman is pregnant, the placenta, which is full of blood vessels, new blood vessels, pumps out FGF1 about 5 times higher concentration in the pregnant woman's circulation. So that pregnant woman for the last 2 trimesters of her pregnancy is exposed to pretty high concentrations of FGF1. And as we know, I mean, there's not a risk factor, at least that I know of malignancy because of a pregnancy.
I'm going to make the closing comments since we're down to the last few minutes here. And what I'd also like to do is I want to clarify something. Vika found out that a pregnant woman has about 5 times the FGF1 in her bloodstream in the 2nd and 3rd trimester. And we're going to be dosing what we think is a very safe level based on everything we've seen in 25 years. But I want to assure all the men, we see no indications that you will get pregnant from this. Ladies and gentlemen, we appreciate your time. All of you have a lot of questions. I've been watching the questions. I've been seeing the stuff go back and forth, and we're appreciative of all the time that you've committed. But let me just give you a couple things real quick. Jack, Vika, and I have been at this for 22 years. We've expended $140 million. And I believe by summer, we will know if FGF1 can reverse brain disorders. I believe the answer is yes. We should know 90 days after the first dosing if the molecule is safe in the brain, which again, I think the answer is yes. And when we know that, then we're sort of cleared to start additional small clinical trials. We don't know if the best dosing is once a day, every other day, every 3rd day. We don't know. What we do know is that we want to find out. So our first clinical trial is for people to be safe. And then after that, we'll start doing larger trials. We'll be doing a trial for people that have DBS so that we can see how it interacts with the DBS. Is there any issues that we can't think of? We'll also do one for people that have had the DBS removed, because we understand they become very difficult situation afterwards. Now, I know that everybody wants to know, how do I get in the trial? What are the standards? We don't know all the standards. We don't know the requirements. We're filing applications in countries where the medical professionals make the decision on the standard for clinical trials. This is made by doctors who deal with the reality of patients and not bureaucrats who deal with their vacation schedule. We may have 20 or 30 small trials going this time next year, and a whole variety of things. Not only ALS, not only Parkinson's, maybe it's tremors, maybe it's stroke, multiple sclerosis. Once again, what we have found in these small Caribbean countries and other countries like Albania and Panama is the doctors say, hey, if it doesn't hurt the people, why don't you do it? Now, we plan on moving as quickly as is possible. And we will not know the inclusion or exclusion criteria until we are told by the doctors in the country that gives us clearance to do the trial. The first question, and the most important question, is it safe? If it's safe, then all kinds of doors open up. But it may be something where the first clinical trial, they say you have to exclude people that are left-handed. And I understand that since I'm right-handed. We are taking people to register for the clinical trials. It's on the website, zgm.care. People are registering there. It'll probably be March before we start figuring out exactly who we're looking for and what the characteristics need to be. Now, let me also make a comment that I brought up a little bit earlier. Besides doing the clinical trials, we need to build up the business of getting this medicine to people. If 30 million people in the world suffer from Parkinson's, and people, whether it's Baku, Azerbaijan, Nigeria, South Korea, Brazil, Argentina, wherever, if we prove the medicine is safe in the brain and we prove we can reverse Parkinson's, it does them no good if they can't get it. So we have to build up that business, that opportunity. And that's why we want to talk to a lot of people who have clinics, whether you're in Morocco or you're in Indonesia, Argentina. We are open to explore if we can do a clinical trial in your country with you and start seeing if we can learn more, faster, and find access. Now, I want to take a step back because a lot of people ask questions or made statements. Why aren't you doing this in the FDA? What's wrong? Why are you running off to all these strange countries? And Jack showed you the ABC News clip where our molecule grew blood vessels in the human heart. We have done 3 trials in the human heart, actually 3.5. I won't bore you with that story. In every single case, it has showed no adverse events. In every single case, the patients have benefited. And yet, in 22 years, we have not been able to get US FDA clearance. It's 20 years since I know the molecule is safe in the heart. It's 20 years since I know it grows blood vessels and saves people's lives. 17 million people died last year from heart disease. And I think we could have extended the life of 30 or 40% had our molecule advanced in 20 years through the FDA. The US FDA, according to Tufts University, takes 28 years to approve a drug. And the cost is $800 million. If you would have come to me and told me 20 years ago that I could go to the Bahamas and be treating humans and saving their lives, we could have saved a lot of lives, people. And I am tired of this game and this ridiculousness where the US FDA approves an Alzheimer's drug that everybody knows doesn't work. In fact, half the panel resigned because it's a joke. And yet our medicine to treat heart disease, the #1 cause of death in the world, is still sitting there. I never would have said this until we discovered that these smaller countries are extremely open. These smaller countries are extremely great. And these countries, we're dealing with a cardiovascular surgeon in Mauritius. And we asked him a question about doing a clinical trial there for the heart indication. And does he need this, the blind pool? Does he need this? Does he need that? Does he need this? All these requirements that the SDA wants from us. And he says, no, I'm a doctor. I can tell if the patient's better. If there's no adverse events and they're better, it's good. Subject closed. So when we found out there's an opportunity to do that with Parkinson's disease, multiple sclerosis, ALS, or whatever, we said we're going for it. Full speed ahead, damn the torpedoes. People are suffering. People are dying. This is not a joke. This is not a game that people can talk to us about. We've met these people. We've talked to you people. We know what the hell is going on when you're sitting there suffering, and all the neurologists can tell you is that you're going to get worse. Well, that's not good enough for us. I believe our molecule will be safe in the brain, whether it's delivered by IV or by intranasal. I believe that as we develop the intranasal, it'll go from a 90-day trial to a 30-day trial, and maybe the time will come where people just pick up the medicine and go home and squirt it up their nose themselves. Once again, the device is a smart device. It won't let anybody make a stupid mistake. That's why I like it. Now, the reason I'm saying to you this is that we understand that there is something more important than big pharma and mid pharma denying people access to medicines. I didn't start in this medicine business. I started on Wall Street where we were cutthroat. And believe it or not, when I was young, I was a real cutthroat competitor. I didn't say it, Jack, but I have never seen a crueler business than this pharmaceutical business. I have seen treatments and therapies out there that I believe could change the outcome, not just for strokes and heart disease and LAS and Parkinson's, but for cancer. And no one proceeds, no one goes any further because it's 28 years and $800 million. Now, we have a webinar next week for ALS. We have a lot of people signed up for it. And you want to know something? We lost a person we love to ALS. We're losing friends now that we love to ALS. They don't have 28 years to wait for the FDA to quit taking meetings with Pfizer to give vaccine injections to 6-month-old children. We have got to go fight. And if there's one thing we are is fighters, folks. So I say this to you because I mean this with all sincerity. We are going to go through the US FDA. We're going to comply with every rule. And if you're in the United States or you're in Britain or you're in Germany and you think that going to the Bahamas or going to Panama or going to Albania is beneath you, fine. Wait the 28 years for our medicines to be approved in your country. If you're an ALS sufferer, we'll see you in the Bahamas. And if you're like many of the people we've talked to where the Parkinson's is ugly, we'll see you in the Bahamas or the Cayman Islands or the British Virgin Islands. We had a Parkinson's trial we were working on with Morocco with Dr. Shahidi. And the application's been there and we were getting close to getting cleared when Jack notified him we want to switch from the IV to the intranasal device. And they said, why? And we said, because if it gets approved in Morocco, a lot of people from Europe will come to Morocco to try the medicine, take it home. Now, ladies and gentlemen, before I say my final sign off, I'd like to say thank you to Bobby. Bobby, I hope you've enjoyed this. You can now have dinner with your family. And Happy New Year, Bobby. We've had a lot of friends, a lot of supporters. We've spent a lot of years at this, but the important thing is I believe the next 12 months, we're going to prove what I've always known. The molecule is safe and it works. If you have questions, mail them to Zhittya@zhittyamedicine.com. If it's a business or finance question, send it to me personally, dan333@aol.com. And if you're really bored, you can email Jack. Ladies and gentlemen, we thank you. We thank you for your patience with us. We thank you for the fact that you have put faith in us. We will notify you about the clinical trials. But there's something my father told me once. He said, never get in a fight. Avoid a fight at all cost. Avoid a fight at all cost. But if you get in a fight, you make sure you win. Ladies and gentlemen, we're going to win this one. God bless you. Have a good day. Bye-bye.